So we're going to continue with some design characteristics. And one of the design characteristics that is the most important design characteristic is your study outcome. So we've talked yesterday about your research design, and your research design is going to be a test of a claim. If always you're claiming that something is superior to something, or not inferior to something, or something is equivalent to something. And so you always have a claim as, and that research question then is going to lead to a type of design, and the design we saw is usually going to be maybe a parallel intervention arms, or we're going to have factorial, or we're going to have crossover, and then sometimes you may not be able to do individually randomized. You may have to do cluster design. So we all talked about that yesterday. And then the next stage that is extremely important is what is the clinical outcome that you're going to use to test your hypothesis. And you are very specific, usually saying, my intervention is going to be better than this other intervention on this thing. And the choice of this thing is what I'm going to talk about now. That is the study outcome. And this is, one of the big problems that we had, and we talked some about it yesterday, is this issue of language, semantics, and that people use things from daily work and daily life in a technical manner when they shouldn't. For example, difference between significance and importance and meaningfulness. We also talked about the fact that when we were talking about who you're going to analyze, we had the intention to treat and modified intention to treat, and what does modification mean? People are not very careful with their words, and they need to be careful with their words. It's a little bit of what Dr. Mahesh was talking about, of being transparent about exactly what you're doing and how you're doing things. So one of the things that we will get into trouble with is people use the word outcome to mean a lot of different things. And I'll try to clarify the difference between a clinical endpoint or a clinical event versus an outcome variable. They're two separate things. You should not mix them all the time. Okay? Then we have complications because many people say, oh, wait a second, I have a primary outcome. I'm going to study, do a trial to see the effectiveness of my intervention on this one primary outcome. But there are all sorts of other interesting outcomes. Those are secondary outcomes. And we will talk a little bit about what if you have multiple outcomes, what do you do with it? And it is a big problem, so much so that the U.S. regulatory agency, the FDA, has a special document on dealing with multiple outcomes. Why? Because this is something that everybody does a lot. Okay? They want it to be done right. We talked a little bit yesterday about the intention to treat and the protocol and that's treated, so I'll skip that. And then tomorrow we'll be talking about the analysis. So this is I'm not going to go through the entire presentation now because analysis will be done tomorrow. Okay, so when people say, oh, I'm going to do the analysis of a trial. What does analysis mean? It means exactly only one thing, or it should be. It's analyzing the primary hypothesis. And your primary hypothesis is always based on a primary question that is your claim that intervention has a causal effect on some outcome. We always do it under the intention to treat principle, especially when it's superiority. It's much more conservative, and it is what we talked about yesterday is what is the way real life is. When a drug or intervention is available in real life in the community, people may or may not follow things perfectly. So even though the trial is an artificial situation, we want some things to be as um, real as possible. So we always do intention to treat principle, which says if somebody randomized their animal. But now comes the outcome variable. And so how do you choose the outcome? Well, this is first of all not based on a statistician telling you this is the outcome. It is not based on what is the cheapest thing that I can do. It's not based on um, what is it that is the most feasible one, but it's really based on what is the most clinically important, relevant thing. And now becomes the question, who is it relevant to? Is it relevant to the physician? Is it relevant to the patient? Or is it relevant to the health service to show that this intervention is good for them? Most investigators think about what is something that I can show works so I can get a publication out of it. That's not the way to choose a primary outcome. It should be something that is clinically important, that in your experience, in your knowledge of the medical community, that field that you're in, it's important to know this because it may change practice if we know this. And practice to be relevant to the patient. This is Dr. Uh, Mahesh said this morning, you have to be always thinking about the patient. You're doing these trials not for you. You're doing the trials to improve the care or to prevent problems in people. You care about them. So a clinical event is something, for example, like a heart attack or a stroke or death or remission or relapse or something that either occurs or does not occur. So it's always a binary situation. Somebody gets diabetes, somebody doesn't get diabetes. Somebody reduces their obesity, BMI to a acceptable level, or they don't. So it's usually a binary thing. That's what an event is. An event is something that occurs or doesn't occur. The word many times used for this is endpoint because that is the event that determines somebody got this or did not get it. Now I don't have to continue to follow that person. So I've ended the follow-up of that. That's where the word endpoint comes in. Now I make a distinction between an endpoint, which is a clinical event, and an outcome variable. So let's take the same clinical event, myocardial infarction, heart attack, that occurs or doesn't occur. I can have different outcomes based on that same clinical event. So an outcome is a random variable, and I could think of, for example, in the 2 years of my study, or the 5 years of my study, or the 3 years of follow-up of people, in the same amount of follow-up time for everybody, did they get that event to occur? Do you remember that big study I talked about yesterday of cholestyramine that set up the cholesterol one? They had minimum follow-up of 7 years of everybody. So in that 7 years, did people get a heart attack or not get a heart attack? That's all they cared about. So that was the outcome variable was the binary variable. Did you get a heart attack in 7 years or you did not get a heart attack in 7 years? So that is calculated as simple as the proportion of people that get heart attacks in group A versus the proportion of people that get heart attacks in group B. Simple comparison of two proportions. It becomes a z-test or a chi-square test. Very simple to do. If your binary variable is your outcome variable. But we could have used a different one. They could have chosen the number. So if you get a heart attack and don't die from it, you can get multiple heart attacks. So I could be counting the number of heart attacks, that non-fatal heart attacks that people get in a given period. So they did not choose that because most people would have died or only get either one or zero or one or two at most, and maybe some people may get three, but they'll either, if you get a heart attack, then things happen to you, right? You get a bypass, and this and that. So then the risk changes. So they didn't use this. But this is used, for example, if I'm interested in diarrhea as my outcome, or I'm interested in something else that can reoccur, like getting the flu, or I can maybe be interested in things that can reoccur as getting COVID again. So in infectious diseases, they use this a lot. In diseases where you can have recurrences, multiple car crashes, as long as they're not fatal and you're still driving, you can have multiple recurrences. So you could count the number of occurrences in a given time period of your same clinical event. Now you can have a count here. You have 0, 1, 2, 3. And some people will have 0, some people will have 1, some people have 2. Maybe somebody will have 20 episodes of diarrhea, for example, in that period. But that's a different outcome variable on the same event. So that would be a count variable. And while binary variables are modeled, are tested with like the chi-square, the count variable could also be tested on the frequency distribution of a count variable, or you could actually use another modeling technique that is the Poisson distribution and be able to compare the mean of the Poisson distribution in one group A versus the mean of the Poisson distribution in group B. And with large samples, the Poisson behaves like the normal distribution, so you can then compare with the z-test. So this is rarely used in chronic diseases because most people will have only 0 or 1, and they will die from it. They want to get another one. But it's used a lot in infectious disease trials or things that can reoccur. And you treat it for the flu, but then you can get the flu again. Or you treat it for a cold, and you get the cold again. They do that in epilepsy, the number of epileptic attacks. So this is another way. So now you can either have a binary variable for a clinical event, or you can have a count variable for a clinical event. Now, binary variables and count variables means that you have to keep observing the person for that entire period and know whether it occurred or not occurred, or it occurred multiple times or not. So you're observing it. Sometimes you have loss to follow-up. People leave for whatever reason, and you lose them. You say, well, wait a second, I only followed somebody for 5 years instead of 7 years. What do I do? I don't know. Do I make up something for those 2 years that are missing? That's one problem. The other one is that I may be interested in not whether it occurred or not, but when it occurred, the time to be occurred. And for example, when not COVID, when HIV first appeared, 1980, 81, 82, there was nothing that, and I gave this example of using placebo in those studies, but once they started, they were trying different things to see what would work. Nothing was working. And if people got HIV, they would go to AIDS, and they would die relatively soon after. So if you did a study and you looked at who dies and who doesn't die under treatment A and treatment B, and you do this follow-up for 1 year, 100% of the people here die, and 100% of the people here died. That's what the initial studies were showing. There was nothing that was working until finally we got AZT. So if you do that, then you cannot compare proportions because everybody gets that. So there is no difference. But some people started saying, well, wait a second, what if we have the people that die here die later than the people that die here? So these people, if we use whatever they're using, or placebo, because there wasn't anything that worked, people die within 3-4 months. But here, if I can prolong their life, even a few more months, that's a success. And so now they were interested in time to death. And if time to death is important, now I'm still, or time to heart attack, then two things happen. I can see if my drug is effective or my intervention is effective in delaying the bad thing, or it's a good thing that I'm looking at in having the cure happen earlier. So now time to that event is what you're interested in. That's one thing that happens. You are changing your research question. Now you're not talking about I'm interested in the occurrence of this. I'm interested in the time to occur. You notice that I'm careful with the words. Now my research question is time to death. So you say, no, my primary outcome is heart attack. No, that's not your primary outcome. Your primary outcome now is time to the occurrence of the heart. Your clinical endpoint continues to be heart attack. That doesn't change. But my variable is not whether a heart attack occurs or not. It's when does it occur. It's time to the occurrence. Now, this kind of variable is called a time to event variable or a survival variable. And Dr. Mahesh was talking about the hazard ratio, and the hazard ratio is calculated by looking at the survival distribution and seeing the rate at which people are getting the heart attacks. And this survival distribution is estimated. Many of you may have heard the Kaplan-Meier curve. That's the Kaplan-Meier curve. It's an estimate of the survival curve. The hazard rate function is how fast is this curve coming down? Is it coming down slowly or is it coming down abruptly? So the hazard is the people that work on life insurance companies, the actuaries, they have a very good term for the hazard. They call it the force of mortality. They work in life insurance, but they're not insuring your life. They're insuring your death. And you don't get anything. The one who gets it is your family. Something. And they know how much to charge you based on your force of mortality. If you are alive this year at the age of 42, what is the probability that you will die within this year? That's how much I will charge you to recuperate money from what I have to pay out for a bunch of people. So I know how much to charge you. If you have a lot of force of mortality, I'll charge you a lot. If you have a very not steep force of mortality, I'll charge you less. And that's what they do. And they constructed these life tables, which are really death tables. And they call them life tables because they wanted to be positive. They would never say, I want to sell you death insurance. People would never buy it. That's why we call it life insurance. But anyway, this is now time to the first occurrence of the heart attack. So now I have the heart attack, and I could study whether it occurs or not in a given time period, the number of occurrences in a given time period, or the time to the first occurrence of a heart attack, or actually, if it's something that can recur, like the diarrheal episodes, or multiple minor strokes, or multiple minor heart attacks, or multiple hospitalizations for heart failure, then I have recurrent events, and I can have now even more complicated ones. I'm still looking at heart failure hospitalization as my outcome. and not whether it occurs in a given time period, or the number of them that I get, or the time to the first hospitalization, but the times to the different hospitalizations. So maybe the first one takes, 6 months, and then after that, the next one will take shorter or maybe longer, shorter and longer, and I have multiple times for different people. This is a little bit more complicated. But the point of this slide is to tell you that you need to be careful of what you said, this is my outcome. Please specify what is your outcome. And keep that separate from this is my clinical event that I'm studying, the clinical endpoint that I'm studying, versus the variable that I'm studying. The random variable is what we statisticians work. So the primary outcome is this one outcome that you think is important, that you think is meaningful to the medical community or to the patient or to whoever, regulatory agency, that is the one you're going to use to test the claim that this is better than that, or this is not superior to that. So it's the one that you're going to use to judge treatment effectiveness. It's the one you're going to design the study around. And you'll see with Dr. J. Seeland, it's the one that the sample size is based. But everybody that does a clinical trial is interested in more than one outcome. Because let's say I'm trying to prevent heart attacks, but for ethical reasons, I need to make sure that I'm not causing chest pain, I'm not causing stroke, I'm not causing hospitalization, I'm not causing any other heart problem, I'm not causing this other thing, I'm not causing not only adverse events, things that you should be also helping at. Then you have the adverse events. One thing is the effectiveness, and the other thing is the harm part. The harm part could be I don't want to be causing renal failure, I don't want to be causing these other problems, or whatever. So we have primary effectiveness outcomes, and we have a bunch of other outcomes. And so what we usually do is call them secondary outcomes. And secondary outcomes are there, and the word secondary should be taken seriously. They're not the ones that you judge your intervention effectiveness on. You're not actually planning the sample size around them. So you may not have adequate numbers of secondary outcomes. Even though they're important, they just didn't occur. It doesn't matter because you did not want to test the hypothesis of the secondary outcome. So we will talk about when we analyze the data that how you analyze this is not any different from that statistically. You'll still calculate the simple issues exact test or the chi-square test or the t-test or the z-test or the log-rank test. Simple statistics. It's all under intention to treat, but you don't really have plans to have adequate sample size to test this. So you should not be putting p-values because the p-value may be not significant because you don't have enough events to be able to test that hypothesis. You didn't plan the study. All you're doing with these is to make sure that you have results that are consistent with the results of the primary outcome. So the secondary ones are there for consistency. And somebody mentioned the word yesterday, exploratory. You're exploring these. So use your words. Oh, we see that we got significant results, positive significant results for myocardial infarction, which is our primary outcome, variable of occurrence or not. And we see that, yeah, our intervention is giving us consistent, small results, but consistent for stroke, for angina, for this, for that, for that, for that, for that. But people love p-values. Put p-values to everything. And journal editors love p-values. They want p-values for everything. And then they start saying, oh, this is significant for that, if that is significant, if there's no difference with this. And that's all the mistakes. Again, Dr. J. Simon said, I said, Dr. Nash said, failure to show superiority doesn't mean that you've shown that you're affirmative. But don't use those words. But people do. So you be careful. Don't do that. All you're doing with the secondary outcomes is looking at consistency of the results. Then you have safety outcomes. Now you look at harm. Are you causing bleeding? Are you causing this anticoagulant that you get? Are you causing something bad? Not, as Dr. Mahesh said, it's not those little simple things like nausea and, vomiting or whatever. Serious problems, bleeding in the brain, or renal failure, or liver damage, or whatever. Those are the bad things. And you also need to assess harm. So the safety outcomes are there to assess harm. Then there is, of course, the economic outcome. Is this intervention, again, you're going to convince people this ought to be used. But if this intervention is one of these injections that you have to take that cost $3,000 per injection, which is ridiculous, who is going to use it? Who is going to be able to afford it? You want to look at this assessing utility. So after a trial is ended, there's a lot of economic analysis of what are the benefits of this, what does this change the practice or not because of economic issues. So there's economic outcomes that people collect data during the trial to be able to do the economic analysis. So those, any questions about the primary outcome? The primary outcome is something that you choose. Don't go after the statistician, what is my primary outcome? Or which one will give me the smallest sample size? That's not the way to choose the primary outcome. Choose the primary outcome of what is meaningful, clinically meaningful, relevant to whoever. Okay. And then you base the sample size on that, and you see, okay, then can I do my study? Is it feasible? Because no, I cannot. Then maybe I shouldn't do the study. If it's not an important one. Maybe one of your secondary ones is you will have a strong effect on that, and you design your study around that one, so now that secondary one becomes the primary, and then the primary that you thought you were going to use would be considered the secondary. So we'll talk more about this when we get to analysis. So many times, yes. So technically correct, because statistical significance is based on what? It's based on having a hypothesis. So you have to briefly specify the hypothesis. And if you have the hypothesis, then you want to test it. You want to test it, you have to have adequate sample size to test it. So if you have multiple secondary outcomes and you want to test them, then you better have made sample size calculations for each one of those that you want to test, and then specify that. And let's say for the primary outcome, you need 400 people, and for secondary outcome number 1, you need 50 people. That's fine. For secondary outcome number 2, you need 800 people. So now your full study has to be 800 people. And most people don't do that, right? They will say, no, I'll just use my 400. And now they'll test their secondary outcome, and they're underpowered to test the secondary outcome. They still do it, and if they're lucky, they get a significant result. If they're not lucky, they get non-significant result, and then they make the mistake of saying, there is no difference in this secondary outcome. That's the problem. So you should not do it. Again, there is no statistical police that's going to put you in jail. And a lot of people do it. But that's the wrong thing to
do. So similar point. So if a secondary outcome is statistically significant, then that would mean inherently that the sample was adequate powered, or they were lucky enough to get.
They were lucky enough.
They were lucky enough, or that sample size taken was enough to show them.
not just the sample size, they got a good effect because the significance is based on two things, right? It's getting that effect, getting low variability. Maybe the secondary outcome is easier to measure with less noise. They get a small effect, but they had adequate sample size. So all three things are playing together in getting that significance.
My question was, if a secondary outcome is statistically significant, I can I can still. It's not wrong to say that okay, this study shows that A is appear to be in this secondary outcome variable that I have.
So
But the question of
it is not wrong to
meaningfulness
say that, but what I'm saying is that you should not base it on whether it has a significant p-value. You should base it on whether the effect that you saw in the secondary one was also meaningful and going in the proper direction. And that the proper direction is consistency of results, and meaningful is your luck that you got because you haven't expected it to be that way. You haven't planned for it. So, yeah, many people get lucky. But if you have 20 secondary outcomes, you may get unlucky in one. And so now you're going to say, wait a second, our intervention worked for these 19, and it's not significant for this, and it's actually going in the wrong direction for this. Now you better explain it. And what people do, they don't even put it in the because they don't want to have to explain. That's wrong. So it's not totally wrong to do significance testing because journal editors love it, and you are trying to please the journal editor. You want to get published. So it's not wrong. I think what is wrong is if you have 40 secondary outcomes, now you choose to report the ones that are the ones you want to report. That's really wrong. But again, as I said, no police is going to come after you for people do.
So corollary to that question, so wouldn't a more effective way to design a study or calculate the sample size be take 5 outcomes that I'm interested in, look at the sample size for each of the outcome as the primary outcome and choose the largest sample size so that you can study 5 outcomes in the same thing. Now the problem with doing a composite outcome is a bit easier.
Yeah, so that's what I'm going to say.
Yeah,
now.
but take 5 individual outcomes that you're interested in rather than choosing one primary. Calculate sample size for all 5. Take the largest sample size so that it will power your study enough to show the significance of each.
Yeah, so many people do that. That's a possibility. You may have to also worry about can you test all 5 of them with independent tests at the 0.05 because these outcomes are correlated. So you cannot even do a Bonferroni adjustment for multiple testing because the Bonferroni adjustment is for independent tests, 5 independent tests. So what most people do is argue what you'll probably be arguing. Because, oh, no, I'm just taking advantage of the fact that I'm doing one large study and testing separately these tests. And if I had done another study with these people and done a paper on them, it would be this. I did another one and used 0.05 here and used 0.05 there. And I could have 5 different papers. I'm just actually studying it in one paper, so I don't have to correct from multiple tests. This is the kind of argument that you do. And depends on the reviewer that you get. They get a reviewer that says, no, we don't buy that argument. So you better correct for multiple tests. So be aware that that may be an issue. But the other problem is, as you said, you would calculate the sample size for all 5, and you will take the one that's the largest. And that one that's the largest may be very large. People don't want to. They will tend to say, oh, let me take one that's in the middle, or I'll take the smallest one because the budget, all of these things require money. And so you will take the one, each one of these 5 is equally important. So I'll just choose the one with the smallest. And then hope, pray that I get significance on the other. But again, significance is not the issue. So if important that you choose your primary that is a real primary. So, but now many people say and argue, I have multiple primaries. And this kind of question arises all the time. And I have it every day when I'm working. The FDA has it also when they get people submitting uh, papers or studies for them, and planning studies. We have different outcomes we're evaluating a new drug on. So they have to deal with this. They came up with guidance for industry, just came out in October of 2022. They finally finalized it. I had the previous draft from 2016, and it is how to deal with multiple endpoints in clinical trials. And this is, you can really download this from the US FDA website and read this, how to handle the multiple outcomes. Now, many people do, as what you said, if I have 4 or 5 that are very important, why don't I do a composite? And composite outcomes are a very attractive thing to do for multiple reasons. Because one of those is I have many primaries. And remember that when you see the sample size calculation, when you're looking at time to an event, especially variable, you need to have a lot of events. So wait a second, if I'm studying time to a heart attack, I may get a few heart attacks. And if I'm studying time to stroke, I may get a few strokes. If I'm looking at heart failure, hospitalization, I get a whole lot of events. But should I get heart failure hospitalization? Well, that's more subjective, and it depends on access to care, this and that. So maybe I should not include it, even though I get more events. But death from heart disease is a hard endpoint. So let me do what the cardiologists call MACE, which is the combination of myocardial infarction, stroke, or cardiovascular death. Hard outcomes, all equally important to one. Why? Because it's time to the occurrence of any one of them. So now I have so many more events. I have much more power. So people do composites for that reason as well, because you get more events, you have more power with smaller sample size. I don't have to have a huge trend. So composite outcomes are very attractive, but people have to be careful what they mix together. So if you put in heart attack, stroke, chest pain, angina, and death, why did you put chest pain? Not that crucial. It's not that bad. Yes, the patient may not like it, but it is not a really terrible thing. It's not comparable to heart attack, stroke, or death. You just put it in for getting more numbers, more frequency occurrence. Same thing with heart failure hospitalization. People like to put it in because they get more numbers, but it is not really comparable to the others. And so you just have to be careful in knowing the clinical importance of the outcomes. And if you're doing a composite, make sure that it is a reasonable composite. I'm not, statistician's not going to tell you what's a reasonable one. Your literature, your clinical colleagues will tell you what's a reasonable. You yourself may know what's a reasonable. But keep in mind that the reason for composite should be that that composite is most clinically important one to study. Not because you get more numbers or you get more power. You should be able to argue this is the more clinically meaningful outcome. That argument is not a statistical argument. So you get greater precision, but what are you mixing? So composite outcomes are a combination of multiple clinical endpoints. They should be equally important. They should have similar event rates. That's why you don't want to put in heart failure hospitalization, because they are occurring very frequently as opposed to a heart attack. and people say it increases the efficiency of the study. Now, there's a difference between having a composite that's made up of three things, for example, or four things, as opposed to saying I have four primary outcomes. Thus, if I have four primary outcomes, then I have to do what you were saying. I have to do a calculation, a sample size for each one of them, and take the highest to do that. And I may be asked to control for uh, multiple testing, or I may not be asked to do it. People may, if they're correlated, there are ways to calculate things. There's also hierarchical testing. So you say, okay, I have these four primaries, but uh, one is more primary than others. And so I will test that one first. And I'll test that one at, let's say I have two. Let's keep it simple. I have two primaries, and I don't want to test them. I cannot test them separately at 0.05. And I don't want to do a sharing of the alpha, as Dr. Mahesh said, that was 0.025 and 0.05. He suggested 0.04 and 0.01. And that's what a lot of people do. They say, okay, I have these two primaries, but this one is more primary than this one. So I will test this one maybe at the 0.05. And if it is significant, then I will say I have not used at 0.05, and I'll test the second one at 0.05. But if it is significant, if it's not significant, then I don't even test this one. That's why we register protocols, and people follow what they say they're going to do. Because otherwise they will do whatever they want. So that's called a hierarchical testing approach. Test the first one at 0.05, and if it is significant, then I test the second one at 0.05. But if it's not, I don't even test the second one. I don't even mention it. That's one way of doing hierarchical testing. And they call that the fallback because it's hierarchical testing. Then they have the hierarchical fallback testing. So I'll test this one at say 0.04, very close critical value at 1.96 will be. I use 0.04. And if it is significant, then I test the second one at the 0.05. But if it's not significant, I still have 0.01 for this one. So I can test the second one. That's called the fallback. So people do these sorts of things. And if you go to this guidance for industry of the FDA, pages and pages of all of these different types of hierarchical testing procedures that the US FDA will allow. Why do they have so many methods? Because a lot of different investigators and especially companies have they have smart statisticians working for them to try to figure out ways to get around doing all of this and testing multiple outcomes at the same time. So they say which thing they will accept and which thing they will not accept. So if you have multiple primary outcomes, that 0.05 must be shared among the primary outcomes. And how do you share it is an issue. They also got really terrible because let's say I'm testing two outcomes and I have two outcomes. And let's say one is heart attack and the other one is stroke. Make it simple. And I think heart attacks is more important for prevention by my drug than stroke. So I'm going to test first the heart attack and then fallback will be the stroke. Okay. Fine, and I'll share the alpha among them. But if I want to call them co-primary outcomes, the FDA has a special definition for co-primary outcomes. So here's another place where you have to be very very careful of your words. Calling them multiple outcomes is very, very different than calling them co-primary outcomes. Why? Because the US FDA says at the end of the day, you're trying to say what? You're going to be able to say, or you want to say, my intervention is efficacious. It works. It's better. And it was based on the primary outcome or on one of the outcomes. So you say, oh, I have multiple outcomes. Maybe you did not get statistical significance for the primary one, but the second primary one, you got it to be. So you could say my intervention is effective because it was good for this second primary outcome. And it is not, did not reach statistical significance for the first one, but it was consistent. And you can say my intervention is effective. However, if you say I have two co-primary outcomes, the US FDA says, fine, you can do that. You can share the alpha whichever way you want. Fine, that's no problem. But if you call them co-primary, you better get significance in both before you say that the study intervention is effective. It's just forcing you to do it. And I've had to correct multiple investigators in our team because it's very sad. Yeah, we have two outcomes. They're co-primary. No, no, no. You cannot call them co-primary if they're going to go through the US FDA. And if the study is something that is not a drug, and it's not going to go to the US FDA, then it doesn't matter what you call it. So this only matters if you are working in pharma and it's a drug, you want to get it to the US FDA. They're the only ones that care about this. You're doing an educational intervention, you're doing Ayurvedic intervention, you're doing this, you're doing that. It doesn't matter what you call it. But you still have to be careful in sharing alpha if you have multiple alpha. That's
I think this is with the five from a general point of view. That case, we have some safety parameters we also have to do. That is small size of network and
so bioequivalent studies are totally different. So all I'm talking about here really does not apply to bioequivalent studies because they are not really bioequivalent. And I'm talking about trials that are going to be submitted to some regulatory agency where you have multiple outcomes that are being studied to get intervention comparison, intervention arm to a comparison. In a bioequivalent study, you have, as you say, multiple parameters that you are estimating. You're not testing. You're estimating what is the maximum tolerable dose, or the rate at which you get to this, how long it stays here, what is it done. You are estimating. And now they are judged differently. They're not judged by, people call them outcomes, but they're not outcomes. Right? They are random variables. They're not based on an event, a clinical event. They're based on a pharmacodynamic or pharmacokinetic property of that drug. So it's completely. You can have multiple things.
because in general conversation, we have also coming the brand behavior studies US and UK. In that cases, we have to try to database upon the some like 95% of the 80 to 95% of the life of the industry. 50 to 175.
know about those generic drugs because we have established a very efficacy already. Only when you generic want to.
So now what you're doing is you are comparing to a reference standard, and that's fine. You are doing a study that is comparing two things, two parameters, then they're judged very differently than these phase two or phase three trials. They're totally different. It's fine to do it as long as the FDA approves the protocol. Now, assume what you do is before you do your study, you go to the FDA and say, here's the protocol. If we show this, will you approve? And then you already have it.
So, because all products have to be guidance. What is your any special?
I don't work with bioequivalence stuff, so I don't actually know the sample size of that, but it would be the if mathematically or statistically you would calculate the same thing. You have to have the mean Pmax. You know, the Pmax is just one value, right? It's not you don't have a distribution of Pmax. Maybe you have multiple people. So you have a distribution of Pmax under your test and a distribution of Pmax under control or reference. Now you compare the two means with the Pmax. You can calculate the sample size you need. I don't know how pharma people have done this, but I'm assuming that if you want to have adequate power, you do the same thing that Dr. J is doing. But now you have a different type of output. Measurement of a parameter, pharmacodynamic, pharmacokinetic. Yeah.
Protocol writing key. Protocol writing. From non-functional could be so as a part of a field plan. How do we train after the study?
So you talk first with a nutritionist. You know what is important from a nutrition epidemiology standpoint. Is it their weight? Is it the distribution of nutrients that they're getting? Caloric intake? I don't know. I'm not an expert in nutrition. But when you talk, again, as I said yesterday, you are not working alone. You're working with experts in the field that know the science of whatever you're studying. If it's nutrition, then you have nutrition people that know. They will tell you what is a meaningful outcome. The study of beauty salons I talked about yesterday, one of the things was consumption of fruits and vegetables as one of the primary outcomes. So why? Because there was a nutritionist on the group that said, you want these people to have healthier lives, healthier lifestyle, then this is one of the important outcomes. So you have to learn from the other disciplines. You cannot guess, right? What should be the thing that
So, the thing that I was discussing about one study. So I want to know whether we can split it into two primary outcomes. The question is extubation cough response. So one is incidence, the other is severity. So if I take it as just as incidence of that cough response, then it becomes very simple study. But then when we include the severity component also, it adds more meaning. So should I take up only the severity if that is more meaningful, or I can take both as primary?
So again, my recommendation is to what is the medical community in your area think is an important thing. The severity is important, and the um, coughing is another one, right? Then you want to do both. But if one of them, if they're both equally important, then they both could be primary. There's nothing wrong with that. Then you have to choose whether one is more important than the other. But that is a clinical decision. That's not a statistical decision. Third thing is then you do, as was suggested there, you do sample size calculation for showing effectiveness of this one and of the other. And if you want to be interested in showing both, then you better take this larger sample size of this. Then whether you share the alpha or not is the other. How you share the alpha. So it's a multiple steps that you go by. But your question, answer to your question is, it really depends on what is you consider more important, or both of them are equally important, or your colleagues consider more important or equally important, or what the medical community or the patient thinks more important. Then you always think about who has to say that this is important. First of all, it's never the statistician. That's the important thing of it. Clinical people that are wanting the study to show that this works for something. That something has to be clinical.
Supposing we decide both are equally important, so what is your suggestion for sharing the alpha, which would be
so if they're totally equally important, then you can be split. But in my experience, most investigators say, well, really, this one is much more important to me, or I think it's much more relevant, or whatever, and I would like to have the opportunity to show an effect on that one more than on this. So you in your mind have that ranking of those two. I try to get it out of you. He's telling me, oh, they're equally, and I will slowly get it out of you and say, but you want severity more than the other. So why don't you give more alpha to the severity one and give less to the other? But give something to the other one because it's also important. You can do the regular hierarchical, which is test the severity first, but if it's not significant, then you don't test the others. But no, we want to do a fallback and have something. So we usually tend to do 0.04, 0.01, or we go 0.03, 0.02, something along those lines. Those are typically what we have done. But if you go here, there's all sorts of procedures. But the important thing is the choice of internally, what do you think? So, mhm. From an ethical standpoint, you must report the safety and the harm. And if you are not causing too much harm, it may be a very simple thing. If you're causing some harm, it can be a very extensive part of your paper, and it should be. Uh, this is ethics that dictates that you must report if there's any harm. So that you have to do. The secondary outcome uh, is not necessarily a must, but it is strongly encouraged because, as I said, the secondary ones are supportive of the primary one. So if you say, oh, I'm studying heart attacks and we prevent heart attacks, and you don't mention anything else, I'm sure a reviewer will say, well, what about, are you causing any other problems? Or are you helping also heart failure hospitalizations? Are you helping strokes? Or are you causing? So the reviewer will demand it, the readers will demand it, so you should report.
most of these trials that you have said that they have objective outcomes. What is your experience regarding when you are approached by a researcher when they are looking at some non-conventional outcomes? For example, when we talk to the statistician, always they are worried, how do we capture it numerically? Sometimes. But for some of the outcomes like happiness or caregiver competence has been intervened along there. So there, when we don't have something to measure that, how do you confront the problem?
So this is a very good question. First of all, yes, statisticians are quantitative science, so we have to quantify one way or another. And often, some investigators are interested in things that are not easily quantified. And it may be things like satisfaction, as you said, it could be fear, it could be happiness or depression, anxiety. And throughout history, research, psychologists have tried to quantify this. And the psychologists have created scales, the depression inventory scale, for example. They've created all sorts of stuff. And many of them argue that this is the best quantification of it. You have to trust those scales, or you don't use those scales. And but if you want to design a study that is a clinical trial, you need to have some outcome that is quantified one way or another, as bad as the quantification may be, or as good as the quantification. So, for example, there are ways that are those inventories and long questionnaires and things. There's visual analog scales where they say, here's your or Likert scales, from totally dissatisfied to totally satisfied, and totally happy to totally unhappy. They have visual ones where they put faces that are going from smile to like that. They keep going along like that, and people circle. And they have all sorts of things like that. We had famous study was this one study of left ventricular dysfunction solved that was done in 1986 to 1991. We were the statistical coordinating center there, and they said, well, people that have ejection fractions less than 50 were the mild ones, and the ones that had ejection fractions less than 35 were the severe ones. There were two trials in one, and they were looking at enalapril as an ACE inhibitor for this. And they said, okay, fine, we can maybe help these people, but how do they feel after? Was one of the researchers, qualitative researchers, no, it's quality of life. And so she proposed lots of questionnaires. But they said, well, how do you quantify it? Said, oh, they had a discussion. So they had the clinical record was about this. The quality of life record was like this, that people had to fill out. And they were asked satisfaction with this and this and that, whatever. And then we're going to analyze it. When we analyzed that, there was so much variability. We did not get really any conclusive results. But there was one question, which was a very interesting one. It was a simple thing called the ladder of life. So here's one. You have a ladder here. 2, 3, up to 10. And they would tell people, where would you put yourself in your quality of life? Are you here at 10? Very good. Or are you here at 1? Just mark. People would go like this. Then we would measure from 0 to here, which is 5.3. Or they would do like this. That was the most predictive thing that worked. 20 pages like that, nothing works. This little simple question. So, some things work, some things don't work. And the way to do this is you have to read the literature. If you're interested in fear, Find out how do people measure fear. You're interested in depression, find out what is the outcome for depression. Read up all of the psychometric properties of these things, which one is has nice convex alpha, look at this, look at that. Work with a psychologist. Find out how things work. Just like he has to work with a nutritionist, you work with a psychologist. Otherwise, we can't tell you that. So, okay. Um,
Same techniques, that is hierarchy testing procedure and hierarchy holdback testing procedure. So both in case of multiple primary outcomes and in case of co-primary outcomes.
uh, yes, yes, you can use them for both. Um, but the only difference, if you can call it multiple primary, you can call them co-primary, and you can do all of these procedures on your study. As long as you're not submitting to FDA, you can interpret it whichever way you want. If you're doing all of this, which you can do, but you submit to FDA, then the only thing you have to be careful is that for the co-primaries, they both need to be significant for you to say my intervention. That's the only thing.
In case if we have calculated sample size separately for all the primary outcomes and then we are taking the sample size, why should we split alpha first of all? Because if we, as you already said, if we run a multiple studies for different endpoints, then we don't do any alpha splitting. But in the same trial, why should we split alpha if we have calculated the sample size for each and every outcome?
So let's take a simple example, right? Suppose I have two outcomes. How about I do this way? We have two primary outcomes. Okay. And so I cannot do 0.05 and 0.05, right? Because then I've increased my type 1 error by having two tests that are usually correlated. So one thing I can do is that fall back, hierarchical, I'll say 0.05. If this is significant, then the second one is tested at 0.05. But if it's not significant, this is not tested. Or I can do 0.04, and I have the other one at 0.01, so that if this is significant, then this one is tested at 0.05, but if it's not significant, this is tested at 0.01. Right? That's the follow. Now comes the other question. I need to calculate sample size. So I better, if I want to test this one at 0.01, where I may end up testing it at 0.01, do I want to have adequate sample size for this? Yes. Then this is going to be a larger sample size than if I have split this 0.025 and 0.025. Because it's a smaller alpha, I'm going to need larger sample size. So splitting it also requires that I calculate the sample size, not for everything at this one, but now at this one. I calculated this one at this and this one at that. And so, yeah, you have to calculate the sample size for all of these. Take the largest for that. This is a problem, right? So people don't really want, they want to have their cake and eat it too. They want to have 6 or 7 primary outcomes and test all of them at 0.05 and then report the ones that are important. You cannot do that. That's why regulatory agencies have come up with these schemes so you don't. So it's not the statistician that's telling you this is a problem. It's because investigators have always wanted to have their cake and eat it too. Do all sorts of things. And the regulatory agencies put a stop on it. They put a stop on it by forcing you to do this. We're just the messengers of the bad news. Don't sacrifice us for the FDA doing this. They're the ones that are forcing you to do it because you are doing it. They're the police. You have to follow that. you may be overpowering the first one. Yep. So then it becomes that you may get significance on the first one because you've overpowered it. And you may, but see, this is where that interpretation of that delta comes in. Because now you'll get such a narrow confidence interval, you're going to be away from zero, but if you're still not even better than your smallest clinically meaningful difference, you better interpret it saying we got an overpowered set. So that was the two decision, two criterion decision, we call it, that Dr. Mahesh talked about. We should also be paying attention so that where we are in what we thought was a meaningful difference. Good question. All right, let's continue. So composite outcomes are very common. People like to use it. In cardiology, which is the area that I work, it's extremely common to reduce sample size and capture. They say the argument is, the clinical argument is we want to capture the overall impact of the therapeutic intervention on all of the cardiovascular kind of problems. That's why we do it. But I know behind all they want is more power. So they increase the event rate, get the statistical power, they reduce the sample size. But caution, they may mislead if the components are of widely differing importance to patients. So chest pain is not as important as having a heart attack. It's not as important as death. Don't mix these two things together in one composite. You get a large number, you may get a significant result, but it may be driven by the chest pain, not by death. What is more clinically important to you? Not the paper being published. What is most important to you should be what is most important to the patient. Death is what the patient cares. Little chest pain is not as important. The number of events of the components of greater importance is small. You may have that you have fewer deaths but more heart failure hospitalization. So you need to know that they are all occurring equally. And the magnitude of effect differs markedly across the components. So when we do a composite, that's our primary outcome. But one of our secondary outcomes is to look at the different components of the composite. Right? So I will do a separate analysis to show if there is consistency in the effect of stroke, heart attack, and heart failure hospitalization. So I will calculate at the end of the day. I may report this is favor. I'll do a little forest plot, favor intervention or favor control. And I will have had a delta that I think is the smallest clinically meaningful one. And I will do my study on the composite of myocardial infarction, stroke, and heart failure hospitalization. That's a composite. So I'll call that composite, which is actually called MACE, major acute cardiovascular event, major cardiovascular event. And I will design my study, and I'm hoping at the end of the day that I get a confidence interval that does not include zero. I can say statistically significant, and that is somewhere like that. What I should be worried about is making sure that I have something that is far away so that there's part of it or enough of it that is above the smallest clinically meaningful one. But then the other thing that I should do also, or most people do anyway, is to get this is for overall, then I will do for heart attacks. What would I get? Hopefully, I'm getting favoring intervention for the heart attack, but I may get it somewhere here, And because I have fewer heart attacks, I may be here. For strokes, maybe it works fantastically for strokes. So I'm over here and I have few strokes, but it's like this. And for heart failure hospitalizations, I may get a bunch of them because that's what's going to happen. So I'm going to have very tight confidence interval for heart failure hospitalization. And hopefully I'm going to be here and I may be like this. All you should be talking about is we have consistent results for the components. Who cares whether it's statistically significant or not? You didn't design the study to have adequate numbers for heart attacks. You designed it to have adequate numbers for this. So why do you put a stupid p-value? You should never put a stupid p-value. You should not talk about significance. The results are consistent if I look at all the components. That's all you should care. Just because these two were significant and this one was not, you don't say there is no difference in heart attack. That is actually wrong. Again, because if you don't reject the null hypothesis, you don't say there is no difference. That's acceptable. This is a very big common mistake that most people do. And I hope nobody does. You need to understand what is happening. Everybody clear on it? All right. So this is this reporting. This is the paper in the I think it was BMJ. I don't know why the J is missing there. Variability, magnitude of effect of interventions across categories of important patients. And looking, they did a literature review. This is not a trial. This is a literature review. And for they looked at multiple studies. This is a, you could call it a meta-analysis, but they're getting multiple meta-analysis that are being presented here, where they're looking at studies that had as an outcome death, studies that had as an outcome critical endpoint, a major endpoint, a moderate, there's a definition of what those are, and a minor one. And these are looking at minor ones would be something like angina, moderate one may be heart failure hospitalization, major may be like a heart attack or stroke or whatever these are over here. You have to look at the papers. And what they're showing is that surrogate outcomes are outcomes that are sometimes used in studies that are because they are easily or sooner observed or easier to observe than the outcome you're really interested in. You may be really interested in preventing, you actually use another outcome, which is lowering of the cholesterol. So you may be interested in studying a stroke, but you look at studying the drop in blood pressure. You may be interested in studying the belly of this horse or doing trials on road safety. You may be interested in preventing road crashes, for severity of people in road crashes, but what you study is speeds lower. Because we all know that speeds are the main and possibly the only modifiable risk factor that will affect crashes occurring and the severity of crashes if they occur. Just like we know that cigarettes are the major risk factor for emphysema, for heart attacks, for uh, lung cancer. We all know these things. Just that nothing happens, right? Because of economic interest. But surrogate outcomes are outcomes that we can measure earlier that are correlated with the outcome that we're really interested in. And so sometimes studies are designed using as primary outcome one of these surrogate outcomes. Why? Because it makes the study feasible. Because I can do it, I can measure it earlier, I can obtain it, and it reflects the potential impact on another outcome. So reducing cholesterol, we presume, will lower mortality due to heart attacks because it has been shown that it is a strong risk factor. So then we design our study not to get heart attacks because heart attacks may be later, and I don't have the time, the energy, or the money to study for 6 years, 8 years, one trial. I am a young person. I want to get moving in my career. I want to have a study quickly. I want to finish it in 2 years. Then let me study something else. Let me study lowering lipids. However, this is a dangerous thing because that presumption may not be true. Yes, lowering cholesterol or LDL cholesterol is highly correlated and is a risk factor for heart attacks. And so, yes, there is a strong association, but it's an association. It's not a if and only if. Right? There is variability. People with low cholesterol may get heart attacks. People with high cholesterol may not get heart attacks. So there is that variability. So it's not always perfectly correlated, the surrogate with the definitive outcome of interest. And so you have to be careful. That's the only caveat I want to mention of the surrogate outcome. So if you design your study on surrogate outcomes, you still have to argue this is an important outcome because the literature shows that yes, if I lower the cholesterol, I will eventually lower mortality or heart attacks or strokes. You have to convince funders of that. Now, this is what makes things complicated. Remember yesterday I said the basic trial design is very simple. You randomize people to A or B, you follow them for observant and primary endpoint, and you calculate the statistic on that, and that's it, you're done. But what happens is people have multiple treatment groups. So if I have now, instead of 2 arms parallel, I have 3 arms parallel. I have control, treatment 1, treatment 2, treatment 3. So I have 3 parallel, 4 parallel. Then do I want to compare treatment 1 with control, treatment 2 with control, treatment 3 with control? Oh, 3, 4 against 3 versus 2, 3 versus 1, 2 versus 1. How many things do I want to compare? Do I want to now adjust for multiple testing and correlated things? I do Dunnex tests, for example, if I was doing like ANOVA. There's all sorts of ways to adjust it. So people have multiple research questions in multiple treatment groups. We already talked about multiple outcomes. But here's another one. People do studies where they're measuring their outcome. Let's say this is one study that we did on looking at cognitive behavioral therapy and dicipramine, which was an antidepressant, women that had functional bowel disease to see their satisfaction with their life, quality of life. And the intervention was 12 weeks of therapy with a psychiatrist. And some people got that, some people got placebo therapy with a psychiatrist. It was an educational thing. They got discipline or not. There was an incomplete factorial design. And after 12 weeks, they asked them the outcome, satisfaction. But the psychiatrist said, you really ought to wait not immediately after the 12 weeks, but 12 weeks later. And they asked them 24 weeks after enrollment. And they said, oh, let's also measure that at 1 year. So now we have 3 time points at which you're measuring the outcome variable. Now we have other things. So you remember I had what I call the clinical endpoint, and then I have the outcome variable. But what if I measure the outcome variable after randomization at T1 and T2 at K times? When am I judging the effectiveness of the intervention? At all times? What if it works at time T1, doesn't work at time T2, works at time TK before? So part of specifying your outcome is specifying your clinical endpoint, your statistical outcome variable, and if you're measuring it at multiple time points, when are you measuring? Is the outcome at 6 months or 24 weeks the primary time at which you're measuring the primary outcome? Or is 1 year the primary time at which you're measuring your primary outcome? You see the difference? You have to specify the time which you measure the primary outcome. That's part of the timing that go into how you describe your study. We haven't talked about this. Oh, yeah, you talked about the PICO. So we have PICO and we have PICOT as well because it has a time. And the timing is one of the timings. So I will, my study will last this long. My recruitment lasts this, my intervention lasts this, my follow-up lasts this, and the time at which I measure the primary outcome is this one, if I'm measuring or observing something. But if it's time to an event, then I don't have to. The timing is during that. So that's why we have multiple time points. That creates a problem. Now, we'll be talking more tomorrow about multiple looks on accumulating data of interim analysis and how to deal with that problem. And one thing that we will also talk about tomorrow is multiple analysis. And remember what I said, that the primary analysis of your clinical trial is ultra simple, right? Because it's adjusted by design, all the potential confounders, it's standardized, it's controlled everything. This is a true experiment. All I have to do is compare the proportions of one, of the effect values, of the differences in means, or a log rank test if I'm doing a survival analysis, unadjusted for anything under intention to truth. Very, very simple. That's the simple primary analysis. But people do all sorts of multiple analyses afterwards. If I have a composite, I'll look at each component. If I have multiple surrogate outcomes, I'll look at that. I will do not only the unadjusted, but I'll do adjusted. What if I had some residual confounding, something that did not work out? Maybe I'll do that. Or, this is what we'll spend more time talking about, people torturing the data so that it confesses to significance in some settings. Why? Because people love p-values that are significant. Why? Because editors love it. And everybody loves it. People do this. And we'll talk about subgroup analysis. If you have multiple outcomes at time points of assessment, this is a calculation of if you start looking more at the data, you have increase in the probability of type 1 error, which is the alpha. If you're doing only one test, it's 0.05. If you're doing two tests, it's 0.10. Three tests, 0.14, because these are the chance of getting significant results with increasing number of tests becomes larger. If you do 10 tests, now you're going to get some sorts of things that are significant. So this is a problem of inflation of the type 1 error. And the danger is that the investigators tend to prominently report things that are statistically significant. Again, trying to please the editors and get their paper published. And so unimportant findings are often prominently reported, or even worse, you may just selectively report what is significant. This is a danger that hopefully none of you are going to do this kind of issue. So if you have multiple outcomes, decide a priori which outcomes are most important. Decide a priori the time period of follow-up if you're going to have multiple time periods. And so it's ideal to have a single one, because that's the one that you're going to do the sample size calculation. Then all other outcomes are second. This is not so simple to do. People have many primary and secondary outcomes, and almost never are trials powered to detect significant differences in secondary. So my advice is always save your alpha for your primary outcome, for the primary analysis of your primary outcome. So if you have multiple ones, then share that among them. But that's the alpha that you have. Everything else that you put in your paper should be talking about describing, exploring what you see. Is there consistency? Is there inconsistency? Is it supportive? If it doesn't support it, but don't use the word significance. Don't put p-values. Do all these other things. You're doing yourself a disservice, and you're doing your readers a disservice. Hopefully, you're not doing the patients a disservice by doing it. So we'll talk about this tomorrow on the integration of intensity tree principle. So I'll stop here.